Hello everybody, welcome back to my channel. So today we are going to talk about enzymes. So firstly we will talk about what is enzymes, then we will talk about effects of temperature on enzymes and the practical for effects of temperature on enzymes. Then we will talk about effect of pH on enzymes. Then lastly, we will talk about practical to investigate the effect of pH on enzymes. So firstly, what is enzymes? Enzymes are biological catalysts. They increase the rate of reaction without being used up. The molecule that an enzyme acts on is called substrate, and each enzyme has its own uniquely shaped active site. The shape that the substrate is complementary to the shape of active site, and this is called lock and key model. So they bond to form an enzyme substrate complex, and once bonded, reaction takes place. Product leaves the active site. So let's talk about the effects of temperature on enzymes. So when the enzyme is heated up to the optimum temperature, it increases the rate of reaction. Because there are more kinetic energy, so there are more collisions, and reaction will take place more frequently. But when the enzyme is heated above the optimum temperature, or it is too hot, it will change the shape of the active site, or we will call it denatured, which is permanent. And the enzyme molecules will no longer catalyze the reaction. So the practical to investigate the effect of temperature on enzymes. Firstly, we put spots of iodine solutions into dips on the spotting tile. Then we heat the starch solution in a water bath, and then we add amylase to the starch solution. Then measure the time it takes until the iodine solution stops turning blue-black, which means that it is the time when the starch is not present, because amylase has broken it down into maltose. So the iodine solution will stop turning blue-black because the starch is not present, because it is already become maltose because of amylase. Then repeat the test with different temperature of water bath. So in this practical, the dependent variable is the time it takes until the iodine solution stops turning blue-black, and the independent variable is the temperature of water bath. So yes, or if you, for your information, if you want to list out some of the control variable, then it may be the volume of amylase or the concentration of starch solution. So let's go on to the effect of pH on enzymes. So enzymes work best at optimum pH. So if the pH is too low or too high, too high or too low, it will change the shape of the active site. Like it is same as having too high temperature. The substrate can no longer fit in and it will be denatured. So yes, it is a bit long. The practical to investigate the effect of pH on enzymes. So firstly, this is things you need to know. Buffer solutions are solutions of salt that resist changes in pH. Then the enzyme catalase break down hydrogen peroxide into water and oxygen. Potato cells contain high concentration of catalase. So, and these are information you need to know before doing this experiment. So, okay, let's go to the procedures. So, firstly, chop a large potato into small pieces and place in a blender with an equal volume of distilled water. Then switch on the blender to mix up the potato tissue and release the catalase from the cells. After that, place 5 cm cube of extract in a boiling tube using a graduated syringe and add 5 cm cube of pH 7 buffer solution by another syringe. Next, mix the buffer solution with the potato extract. Then leave the mixture for 5 minutes. Okay, then add 5 cm cube of 5% hydrogen peroxide solution to the tube from a third syringe. Next, we insert a bung and delivery tube in the boiling tube quickly, and the end of the delivery tube is placed in a beaker of water. After that, count the number of gas bubbles produced in the first minute after adding the hydrogen peroxide to measure the initial reaction rate. Lastly, repeat the experiment using different buffer solutions. So for the dependent variable in this experiment, it is the number of gas bubbles produced in the first minute after adding the hydrogen peroxide, and the independent variable is the different pH of buffer solutions, and the control variable is like using the same potato, and the concentration of hydrogen peroxide and the volume of hydrogen peroxide. So yes, this is the end of the video. I will put the notes of this lesson in the description below. Please like and subscribe and share this video, and yes, have a great day and bye bye.